Bismillahirrahmanirrahim. The transcription in eukaryotes is far more complicated as compared to the transcription in prokaryotes. Eukaryotic transcription involves separate polymerases for the synthesis of ribosomal RNA, transfer RNA, and messenger RNA. In addition, a large number of proteins called transcription factors which bind to distinct sites on DNA within the core promoter region are involved. These transcription factors influence the assembly of transcription initiation complex at the promoter and the determination that of which genes are to be transcribed. Each eukaryotic RNA polymerase has its own promoters and transcription factors. The association of DNA with histones to form nucleosomes affects the ability of the transcription machinery to access the DNA to be transcribed. Most actively transcribed genes are found in relatively decondensed form of chromatin called euchromatin, whereas most inactive segments of DNA are found in highly condensed heterochromatin. And interconversion of these two forms is called chromatin remodeling. A major component of chromatin remodeling is the covalent modification of histones. For example, the acetylation of lysine residue at the amino terminus of histone proteins by histone acetyl transferases, which eliminate the positive charge on lysine, thereby decreasing the interaction of histone with the negatively charged DNA. Removal of the acetyl groups by histone deacetylases restores the positive charge and restores the interaction between positively charged histone proteins and the negatively charged nucleic acid DNA. These enzymes are able to access DNA in presence of ATP, which allows repositioning of DNA, exposing the lysine residues, which may then be acetylated or deacetylated. The template strand is the non-coding strand and the non-template strand is the coding strand because the transcribed RNA is identical to the non-template strand. During translation, messenger RNA is read 3 to 5' in sets of 3 bases called codons that determines the amino acid sequence of the protein. Thus, the base sequence of the coding strand of the DNA can be used to determine the amino acid sequence of the protein. For this reason, when gene sequences are given, they refer to the coding strand. There are 3 types of RNA polymerases in the nucleus of eukaryotic cells. All of these are large and contain several subunits. Each type of RNA polymerase recognizes specific genes. Since mitochondria represent the prokaryotes which were engulfed by eukaryotes during the process of evolution, the mitochondria contain a single polymerase that resembles the prokaryotic RNA polymerase. Eukaryotic RNA polymerase synthesizes the precursor of 28S, 18S, and 5.8S ribosomal RNA in the nucleolus. The eukaryotic RNA polymerase II synthesizes the nuclear precursors of messenger RNA, which are processed and then translated to proteins. RNA polymerase II also synthesizes small nuclear RNAs involved in RNA editing or post-transcriptional modifications of the RNA. In some genes transcribed by RNA polymerase II, a sequence of nucleotides TAT AAA, that is nearly identical to the Pribno box of prokaryotes, is found. It is centered minus 25 nucleotides upstream of the transcription start site. This core promoter consensus sequence is called the Tata or Hogness box. However, Tata box is not present in majority of genes. Instead, different core promoter elements such as INR or initiator sequence or downstream promoter elements DPE are present. It is important to appreciate here that not one consensus sequence is present in all promoters. Since the consensus sequences are present on the same molecule of the DNA that is being transcribed, so these are called cis-acting elements. These sequences serve as binding sites for proteins called general transcription factors or GTFs, which in turn interact with each other and with the RNA polymerase II. General transcription factors are required for the recognition of the promoter, recruitment of the RNA polymerase II to the promoter, formation of the pre-initiation complex, and initiation of the transcription at a basal level. Since GTFs are synthesized in the cytosol and move to their site of action, therefore these are called transacting. In contrast to the prokaryotic RNA polymerase, the eukaryotic RNA polymerase or the RNA polymerase II does not recognize and bind the promoter by itself. Rather, transcription factor TF2D recognizes the promoter. Another GTF TF2F escorts the RNA polymerase II to the promoter, and yet another GTF, the TF2H, with helicase activity, melts the DNA to be transcribed, and its kinase activity phosphorylates polymerase II, allowing it to clear off the promoter. Not only the consensus sequences are found in promoter region, additional consensus sequences called regulatory elements are also present. These are special DNA sequences that not only influence the frequency of transcription but also the response to signals that affect transcription and subsequent protein synthesis. These upstream regulatory elements may further be categorized as proximal regulatory elements, which are present within approximately 200 nucleotides, or the distal regulatory elements, which are present farther away from the promoter. Special transcription factors or proteins called the specific transcription factors or STFs bind the proximal regulatory elements, for example, STF, CTF, and SP1, binding to the proximal regulatory elements CAT and GC boxes, respectively, and influence the frequency of transcription. Still, other STFs bind distal regulatory elements like enhancers to mediate the response to hormonal signals for transcription. So, here you can see the STFs binding to the CAT and GC boxes. STFs have two binding domains. One is the DNA binding domain and other is the transcription activation domain that recruits the GTFs to the core promoter, as well as the co-activator proteins such as histone acetyltransferases or HAT involved in chromatin remodeling. Mediator is a multi-subunit co-activator of RNA polymerase II, and it binds the polymerase, the GTF and STFs and regulates the transcription. The supercoils formed during the process of transcription are removed by the same topoisomerases that remove supercoils during DNA replication. Enhancers are special DNA sequences that increase the rate of initiation of transcription by RNA polymerase II. These are typically on the same chromosome as the gene whose transcription they stimulate. However, they can be located upstream towards the 5' site or downstream towards the 3' site of the transcription start site. These can be located close to the promoter or maybe thousands of base pairs away from the promoter. These may be present on either strand of the DNA. Enhancers contain DNA sequences called the response elements. These can bind STFs. By bending or looping the DNA, STFs can be bound to an enhancer sequence at one point and to the GTFs bound to the promoter and the RNA polymerase at the other point. Mediator also binds the enhancers. Now, silencers are the DNA sequences similar to enhancers in that they can act over long distances, but they reduce gene expression. A strand of DNA that serves as template for DNA synthesis is copied in the 3' to 5' direction. Synthesis of new RNA molecule occurs in the 5' to 3' direction. The ribonucleotide triphosphates ATP, GTP, CTP, and UTP serve as precursors. Each nucleotide base sequentially pairs with the complementary deoxyribonucleotide base on DNA template strand. Adenine base pairs with thymine on template strand. Guanine base pairs with cytosine on template strand. Cytosine pairs with guanine on template, whereas uracil joins adenine on template strand. So here you can see that pyrophosphate is released and guanine moves to base pair with cytosine. The eukaryotic transcription is far more complicated process than transcription in prokaryotes. Not only the eukaryotic transcription involves separate polymerases for the synthesis of ribosomal RNA, messenger RNA, and transfer RNA, but a large number of proteins called transcription factors are also involved. Compared to one circular chromosome of prokaryotes, humans have 23 linear chromosomes per haploid cell associated with histone proteins which are absent in prokaryotes. In prokaryotes, not only the RNA is polycystonic, the translation and transcription take place simultaneously, whereas in eukaryotes, the RNA is monocystonic and the transcription and translation are not coupled.